So it is a great pleasure and a great honor to introduce our next speaker, Professor Luc Montagnier. Professor Montagnier, of course, is best known for receiving the Nobel Prize for Physiology or Medicine in 2008 for his discovery of human immunodeficiency virus, the causative agent of AIDS, one of the greatest medical breakthroughs of the last 50 years or more. But today, he's going to talk about his equally groundbreaking work, which concerns homeopathy, informative water structures in diseases, from quantum physics to homeopathy, and indeed, Professor Montagnier was a close associate and a colleague and a friend, I believe, of Jacques Benveniste. So here he comes, Professor Montagnier.
Thank you very much for this invitation. Actually, I've been participating in several conferences on water, and I think it's first time we seem to have some kind of convergence between the different presentations from quantum physics, from chemistry, and from biology. So I am optimistic in this way, but as I will show you, we are also meeting some new problems of our humanity which may interfere with our own life. Actually, I will start by telling you a story. A French journalist, his name was André Frossard, a journalist of a newspaper now which has disappeared in France, but he has his glory time in the beginning of the 20th century by producing a paper titled J'accuse, I am accusing. And this was written by a French writer at that time, Émile Zola, and it was the beginning of the Dreyfus affair. We have now also a similar affair, but we hope that this will not put people in jail for years. And this journalist, André Frossard, wrote a book saying, God exists because I met him. I'm not going to say that today. I didn't meet God yet, perhaps, but I met homeopathy, and I can tell you it does exist. So I'm, of course, basing this on scientific research, perhaps also some personal experiments, but I will not talk about my personal problems today, which are not very important. But I like to show you the basic, what I think, the basic basis of scientific research showing that homeopathy does exist in nature before also used by men, but also existing in nature. And this goes to a coherent work between physicists, doctors, medical doctors, and biologists. Next please. Next slide. And of course, you won't be surprised that we also involve water as the main, not only the main memory things, but as the organizing of life, and also organizing our disease, because not only us are using those principles, but before that, also microorganisms discover it. And actually what we are fighting many chronic diseases in which some new structure of waters are involved, but not coming from our own DNA, coming from the DNA of our surrounding parasites. So we should not forget that nature has already started the fight, and we should continue it, but it is a long-term fight. Next, please. So we started by a very simple model of biology, of molecular biology, called PCR, polymerase chain reaction. What is it? Is it a system in which we can detect specific DNA sequences by using first primer, small oligonucleotides, about 20 nucleotides long, which could be the origin of a synthesis by a very unusual polymerase, which is the Taq polymerase. A polymerase extracted from Thermophilus aquaticus bacterium, which is living in hot spring in the United States. And the discovery was made by Carey Mullis in the 80s, and he received rightly the Nobel Prize for this discovery, because it can be used for amplifying millions of DNA molecules by chain reaction, because this polymerase will work first on open strand, opened by heating at the high temperature. And this first start then will not destroy the polymerase. The polymerase is heat resistant. And this allows the polymerase to reconstruct again a double-stranded DNA, which could be opened up again by heating and so on. So this is a really chain reaction, not atomic chain, but chain reaction, which could be used to amplify millions of DNA molecules. And the first question we asked actually was a theoretical question to physicists, to our quantum field physicists, was how the recognition of the very specific DNA sequence by those oligonucleotides can be made in a random system, in a Brownian random system. And they told me that perhaps there will be an intermediate in this recognition, which will be a resonance intermediate, waves. And so this is why we started by publishing a paper in the journal Water, edited by Jerry Pollack, on the possibility of that polymerase chain reaction should be more complex, involving a third party, which should be water structure. Next slide, please. We started with a very simple system, well known in the pathology, unfortunately, is the Lyme disease. And Lyme disease is caused by a polymorphic bacterium, but can be also, unfortunately, involved in chronic infection. This is controversial, but I think many patients, and more patients will tell you this is real, the reality. Neurological infection and arthritis, oxidative stress, many signs of chronic infection are there. And the question was, is the germ, the Borrelia bacterium, involved in all those symptoms? I think molecular biology will tell you yes, and especially PCR. PCR can detect DNA in the blood of patients suffering of chronic Lyme disease. We did some work some years ago on that. Not only us, but several other groups as well. Next slide, please. So this slide, this electron microscopy slide, show you this is a scanning electron microscopy study, which showed that the high polymorphism of this borrelium, which is like the Treponema pallidum, cause of syphilis. And I think Lyme disease will probably have the same importance in our century than syphilis in the 19th century. The difference is syphilis now can be cured by penicillin, and penicillin is derivative. This is not the case of Borrelia, which is resistant to this kind of antibiotics, and actually is sensitive to several types depending on this morphology. So between the spiral form, there is also a globular form of Borrelia, which can persist in several tissues for a very long time. But I will show you also that there is another form of Borrelia which is derived from water, another form of DNA which is derived from water structure. So the fight of medicine is just beginning against this kind of chronic infection. Next slide, please. We design, like others, some specific polymers, oligomers, recognizing the ribosomal DNA. Actually, this germ not only has its own genomic DNA, chromosomal DNA, one chromosome, but also many plasmids, some form of DNA replicating independently from the genome. So it means in order to detect broadly the whole kind of Borrelia, we focused on the use of ribosomal DNA primers, because ribosomal DNA belongs to the genomic part of this bacterium. So this was designed by one of our collaborators, Dr. Lavallée, for some time ago. And we also designed what we call nested PCR, which recognize some internal part of the first DNA recognized by some oligomers. And so actually our classical test was the use of primers for detecting one part of the 16S ribosomal DNA, which had 499 base pair. And by amplification by 40 to 55 cycles, we were able to detect by classical PCR some DNA in some patients suffering from chronic Lyme disease, but not all. Next, please. So this slide shows the results of one classical PCR on the left, showing this band of 499 base pair, which, when we were sequenced, showed exactly the sequence of Borrelia burgdorferi. But unfortunately, there were cases in which we could not detect that DNA by classical PCR. Next. So here comes the homeopathic approach. We were using dilution. Thank you. We were using high dilution and see if we could detect that DNA by PCR in the high dilutions. High dilution do not contain any more DNA, Borrelia DNA, but they were taken from patients suffering from that disease, and indeed we could detect Borrelia DNA by dilution, high dilutions in water of their plasma DNA. Next. So the first step of this new technology, which is called ultra PCR, because it goes beyond the one DNA molecule of DNA, was to dilute out in 10 times, tenfold dilutions, starting at very low concentration, either of first a control Borrelia DNA, 2 nanograms, but also starting with the DNA from the plasma of Lyme disease patients. Next, please. And this was theorized by a very close collaboration with a group of physicists, quantum field physicists from Italy, and we're working with some American colleagues. And we published last year in the journal Water this paper, which was really intriguing, water bridging dynamics of PCR in the gauge theory paradigm of quantum fields. Actually, this was an explanation of the effect of ultra PCR by showing that there was really an intermediate between the enzyme, the polymerase, the Thermophilus aquaticus, and the supposedly existing structure of DNA-like existing in the plasma of patients, in the blood of patients. Next, please. So this scheme from this paper showing the principle of the explanation. On the left, you have the DNA double helix. Of course, the shape, the size of those three structures is different. So you have the double helix, the beginning of the one double helix. On the left, you have in the middle small birds flying, but all in the same direction. This is supposedly the activated water molecule, like photons in a laser. They are existing as dipoles, but they are also existing as a domain, a coherent domain, which contain millions of water molecules in a different state, perhaps in the same activation state, and this could fit with the idea of a charge electron of charge of the structure, which have the size of about what has been described by the previous speaker, Jerry Pollack, Vladimir Voykov, of about 100 nanometers. Why is that? Because we could filter the system by filtration and retain the structure by 100 nanometer filters. At 20 nanometer filter, we could decrease the structure but not completely abolish it. So this means there is a variability of size of those structures. On the right, you have a scheme of the protein structure, the peptide structure of the Taq polymerase. Again, this Taq polymerase is a bacterial enzyme, but as you will see, it has the property not only to read directly the base sequence of DNA, but also the supposedly informative structure of water informed by the base sequence, but not working on the DNA helix itself. So it means we can dilute out this water structure for many folds and still keep the information of the DNA. Actually, this is something new in the field of homeopathy, because in this case, we do not show some imitation by water of the function of one specific chemical or biological molecule, but we have the possibility to read exactly the high polymeric structure of DNA, its base sequence, because here we can directly sequence the product and see if it is exactly the product of DNA, the sequence of DNA or something else. So this is a three-parted system, more complex than what is already thought. But as you will see, we use this hypothetic model to do the experiments, and the experiments validate exactly the hypothesis. Next slide, please. I forget to mention that there is something very important, concussion. Concussion has been used in homeopathy from the beginning, and it is indeed a real need, a necessary step of having the system read without the DNA molecule is to agitate, strongly agitate each dilutions. That is very critical. So we will show you now some results which would indicate that we can read some water structure having retained the sequence of a DNA. So it's not only showing a function, an imitation of function, but it is also a transmission of genetic information by water. Next, please. So when we dilute out, of course, we can suppose that, according to the previous theory, that the water structure, which will retain some changes, which will be read by the Taq polymerase as DNA, as base pair. So we will each time do some experiments in which we control the sequence which is produced by this unusual system. PCR of DNA without DNA, homeopathic. Next. So this is just a control experiment in which we use the Borrelia DNA, amplicon, having in the base sequence I said previously, and different dilution. So this slide is actually electrophoretic gel, in which on the left you have the beginning of the dilution, -2, -3, and after that, at about the dilution 7 times 20, 10-fold dilution, you see again some DNA bands at the same size of the DNA bands of the controlled DNA. Of course, in this kind of experiments, which is highly sensitive, you have to be very careful. And the first is to avoid contamination, of course. We have to design a special laminar flow hood system, like working on dangerous viruses, to make sure there is no transmission from one sample to another. Knowing that at each time you have to shake for 15 seconds very strongly with a vortex system. This is the concussion step. And you see bands of DNA at the same size, even at 20-fold times dilution. At 20, again, starting from -7, there is no calculation indicate there is could not be any DNA molecule left. So this implies that the reading is done on something else in water, and the something is exactly the same sequence as the originating DNA. So this has been checked by, of course, sequencing of those bands, and again, the sequence was exactly the same, one plus or minus one nucleotide. Next slide. We then went to patients, Lyme patients. And this person, as you can see, you don't see very much on the left, the DNA itself, because this is also very important. The enzyme reaction was done without DNA extraction. It means we could read some DNA structure inside the plasma of that person. And of course, at the beginning of the first dilution, you have a mixture of many things in the plasma, which could be inhibitory of the PCR reaction. So what is important is what you see on the right. You see some dilution which are negative, and this is a characteristic of homeopathic studies, have been shown also by Vladimir Voykov for chemicals. You can see some holes, some DNA, some dilution in which DNA cannot be detected. It doesn't mean the information is not there, because if we took this negative dilution, it will transmit the information to further dilutions. This is very important. So something exists, but we cannot detect it here for reasons which are obscure now. But I think this is the real thing, and this has been shown also for the transmission of functional molecule by Jacques Benveniste and his colleagues. But here you see a region in which there is a continuous presence of DNA bands at dilution starting at -14 and up to 20. We stop at 20 because this is a very boring, time-consuming experiments, but we hope to make it robotized, and this will be done by machine in a more easy way, in more also way to avoid contamination. Each time, of course, we do the control with the water which not been agitated, to see if we could detect something, and the answer is generally not. Next slide, please. This is the case of the same family, but this is the mother of this boy which had recognized Lyme disease. He was also positive by classical PCR. But in that case, in the case of the mother, she was completely negative by classical PCR, indicating this system can be used also for diagnostic amplification, amplifying the possible presence of Borrelia DNA. Borrelia DNA is there, not always, but in most of the dilutions. Next slide, please. This is another case of another patient in which we see again many dilution positive, but some dilution also are negative. So for this time being, we cannot quantitate this system, but it can tell you yes or not, Borrelia DNA is involved or not. Next slide. So we discussed what could happen in those very curious dilutions. We think, as has been said also before by the previous speakers, that there are nanostructures, he called them nano-associate at the Russian way, I call it at the French way, nanostructures, which goes a little more precise, but still opening up many possible hypotheses. Those nanostructures can carry the DNA information. This is the first time we can demonstrate that dilutions are informative. The dilution water has found a way to carry very precise information, which is the base paired sequence of DNA for about 500, at least 500 base pairs. Next slide, please. We call that ultra PCR so far because it could be applied to other chronic disease. And this is, I'm not just saying, but talking about this first part, showing that this, in the case of Lyme disease, it could help detection of a chronic infection at a very dilute systems. But what we are doing is to start from plasma DNA, and it's quite possible that even without dilution, there are some structures which could play a pathologic role in the blood of those patients. Next, please. So this slide summarized the step of this ultra PCR. starting with human plasma, prepared under laminar flow hood, class 100 hoods. No DNA extraction. Filtration is important to remove bacterial DNA body and so on. And then serial decimal dilution or centenimal dilution, what the homeopathic people call CH. It works also with CH dilutions. Up to here to 10-20. Vortex agitation is very important. Concussion. Next slide. Then PCR on each dilution with the primer for 16S ribosomal DNA. We separate the DNA bonds by classical agarose gel electrophoresis. We stain with fluorescent dye. Eventually, we clone that DNA band to have a sequence in bacterial amplification. And this gives you a diagnostic of presence of those structures. It doesn't mean we don't know exactly their role in the disease, but it means there is some live DNA, genetically active DNA, in the blood of those patients. Next. So this cartoon will show you what we think. Actually, homeopathy does not make high dilution. There is no such thing. Each dilution, according to perhaps the most simple idea of liquid crystals. There are liquid crystals carrying this information in the water. And if you dilute out 10 times, at the beginning, the tube in the low blue will not contain as much as liquid crystals, but concussion, agitation, will reproduce the existing liquid crystals, all the previous dilution. So each dilution had the same, finally, liquid crystal concentration and same information. But we, of course, have to explain the holes. Some DNA, some dilutions are negative, we don't know why, but they are transmitting the information to the next dilution. So homeopathy does not dilute. There is no such thing as dilution. Actually, there are maintenance by mechanical shaking, apparently without electromagnetic field, but I did not discard. We did some previous work in which you could transmit some information, DNA information by electromagnetic signals. But here, to show for the people which are completely ignorant and do not believe in electromagnetic fields, then we show them just by dilution and shaking mechanically, we can reproduce the original concentration. So I think this is a clear demonstration that homeopathy works. But of course, there are many parts of the theory which need many studies. How the information for DNA, for chemicals, for biological products can be kept in a system in which there is only water, apparently only water. We cannot discard the possibility that in a system there are impurities which play a role. So I don't think there are, we'll see now, there is no such thing that pure water. Pure water does not exist in our world. We are not made of pure water. We are made of water, but not pure water. Next slide, please. I'm now going to turn to another pathologic system, another disease, which is autism. In 2012, based on the work of our medical colleagues, which used antibiotics to treat early autistic children, And with some success, they could reduce the sign of autism by this treatment. And as we had at the same time some indication that electromagnetic signals also were present in those patients, indicating perhaps bacterial infection. We tried to publish the infectious tract of autism. I first spoke about this in a Chicago meeting of Autist One Society without mentioning anything but infection. No vaccine the world vaccine was not used. But since I was participating to a meeting in which there were parents which had some concerns by some effect of vaccine, I was put in the catalog of anti-vaccine. The title of Forbes was Nobel Prize joined the anti-vaccine crowd. This was in 2012. And after this meeting, also, I organized at the French Academy of Medicine, not the Royal, the French Academy of Medicine, a meeting on that, showing mostly the data obtained by clinicians. And this meeting was well accepted but not published. The academy authorities did not authorize the publication of this paper, saying that we had showing our data, just possible infections, bacterial infection could lead to autism. Next, please. And now we are in 2018, 6 years later. Autism has really become a very important problem in the United States and also in many developed countries. And you can see here the latest CDC figures. CDC first tried to hide the data, but they now recognize the real situation. And we have to explain this increase. I think this is like an epidemic. More and more children are hit by autism or autism, what we call autistic spectrum, which involve several symptoms of autism. At the same time, in 2012, next slide please, there was a paper published, not by us, by the group of Jan Lipkin, which was a very well-known group for hunting viruses or bacteria in new disease. So they applied their skill to the detection by PCR, 16S DNA, of some bacteria taken from biopsies of children suffering of gastrointestinal symptoms of autism. So this was well organized in their lab, and they published a paper, which you can read here, showing that in a group of children suffering from gastrointestinal symptoms, there was a change in the flora detected in biopsy of the ileum, the final part of the small intestine, and those children compared to a group of controlled children suffering from similar symptoms but without autism. This was published in 2012. At the same time, we were trying to publish our own results. We are now in 2018, and there have been no further publication supporting this or affirming this in the literature. Why? This is an open question. But we were not accepting the silence. And we applied the same primers for 16S DNA of this new group called Suterella to the study of the plasma of children suffering from autism. First using the classical PCR, but after that, of course, using the new ultra PCR, which was considered more sensitive. Next slide, please. So we applied this study on a group of children suffering of autism, and we were amazed by the results. But not only we could detect a group of bacterial DNAs of several bacteria groups by this technology in the patients suffering from autism, but everywhere. In the water we are drinking, in the blood of patients not suffering from autism, but suffering from neurological disease, in some biological preparation. I will not mention all of them because this will be considered some attack against products made by the pharmaceutical industry. I am not accusing nobody at that stage because the discovery was this positive reaction existed almost in every food we can eat, including the milk of course, for the babies. So it seems that, I'm not saying this is the cause of many diseases at that time, but it seems that we are now in a world in which some bacteria, for some reason to be discussed, have almost exploded in our world. Next, I think, show you, I don't know if you could read it, but most of those bacteria are still very specific because if we use the primers of Lipkin, we detect bands at the right size, and they belong to selected groups of bacteria, mostly the Burkholderias order. And in that order, we detect, of course, Suterella or Parasuterella, but also some other group very close, like Achromobacter, Restonia, Herbaspirillum, bacteria we haven't heard too much. We ourselves haven't heard too much before. Where are those bacteria? They're coming from the soil and from perhaps the food given to the animals we are growing up for us, for meat. So this is an open question, but which deserves further research. It's quite possible that because of the change of our environment, especially the changes occurring in animals fed for us, cattle, for instance, chicken and so on, that there has been a change in the microbiota of those animals. So they are now contaminated by this kind of bacteria. Some of them were known to be weakly pathogenic, especially immunocompromised. They were being found in immunocompromised patients. But the problem also, they have been found in reagents used for PCR. So it's quite possible that in some cases, and we are trying to solve that problem, of course, that the water we are using for dilution are already contaminated by reagents using for PCR. We are exploring now another world in which those bacteria not only exist, but they have been spread in not only in the animal population, but also in the human population. So everything has to be looked for. But we cannot discard the possibility that we are dealing with some bacteria, which in some cases are pathogenic, especially for chronic neurological diseases, like stroke, for instance, and multiple sclerosis, and so on. So it's quite possible that we have to look for those bacteria. It's not only possible, it's necessary to look for those bacteria in those diseases, but also in our food, in nutrition. And of course, this applies perhaps to modify our way of consuming animals, animal meat, and perhaps also even being vegetarians. So this may have important consequences, not only in medicine, but in agriculture, in what we call industrial food, industrial feeding. Feed is not very well, it's not a very good word, but maybe we are exposed to feed and not to food, especially. So this implies some reflection, some information first, some studies, some research studies, but you can see a very different picture than having a disease with a specific bacterium as the cause. Here we are dealing with something very broad, and simply I think it's very important to apply the principle of precaution. We don't know, but there is a possible danger, so we should avoid anything which could cause the increase of that danger. So I will finish by showing you a few slides of reflection. Next slide, please. You can see, for instance, this study shown by Dr. Stephanie Seneff from MIT, which compare the consumption of glyphosate in thousands of tons and the increase of autistic children number. The curves are parallel. Doesn't mean one is the cause of the other, but it needs, some studies. Can we stop? Can we use other Roundup like herbicide, which have not the same effect as this herbicide, which actually is very close to an amino acid glycine and could be incorporated in our proteins. Who knows the long-term effect? Nobody. Next slide, please. Also studying the change in the barrier, which are physiological, between our gut and blood, and between blood and our brain. I think this is very important, perhaps in the young years of life. Next slide, please. Show you possibly some changes in the opening of those barriers, especially from gut to blood in young babies and in aging people from blood to brain. Next slide, please. We have to consider that we are dealing with, chronic disease with multiple factors, chemical factors, but also factors brought by medicine itself. Vaccines containing adjuvant, strong adjuvant like aluminum, which could have some effect on autoimmune disease later in life. We have to deal also with the electromagnetic surrounding, which have immensely been increased in the last 10 years. And we don't know where we are going to where. Is it something safe or not? We have to be very careful and make independent studies from the producer of those, which are, of course, very economically important now. next. And my last conclusion is that health should be over economy. This is really important for our own future and the future of the next generation. Thank you very much.